proteins like GDF11 as potential pro-youthful factors, while others like CCL11 have been pegged as pro-aging. The ultimate goal is to create a recombinant cocktail of beneficial factors or to develop drugs that can block the harmful ones. This would provide the potential benefits of parabiosis without the logistical and ethical complexities of blood exchange. We now arrive at what may be the most futuristic and potentially transformative frontier in all of aging research: epigenetic reprogramming. To understand this, we need to distinguish between genetics and epigenetics. Your genetics, your DNA sequence, is like the hardware of a computer. It's fixed. Your epigenome, however, is like the software. It's a complex system of chemical marks and tags on your DNA that tell your genes when to turn on and off. This is what allows a skin cell and a neuron to have the exact same DNA, but completely different structures and functions. A leading theory of aging posits that a primary driver of the aging process is epigenetic drift. Over time, due to environmental insults, metabolic processes, and errors in maintenance, this epigenetic software becomes corrupted. The precise patterns of gene expression that define a youthful functional cell become noisy and disorganized. Genes that should be off get turned on, and genes that should be on get turned off. This loss of epigenetic information is thought to be a fundamental cause of cellular identity loss and functional decline, which we perceive as aging. The revolutionary breakthrough in this area came from the work of Dr. Shinya Yamanaka, who discovered that a combination of just 4 specific transcription factors, now known as Yamanaka factors, could take any adult cell, like a skin cell, and reprogram it all the way back to an embryonic-like pluripotent stem cell. This was a complete reset of the epigenetic landscape. While this is incredible, a full reset is not desirable for an anti-aging therapy, as it erases cell identity and can cause tumors called teratomas. The key question became, can we partially reprogram cells? The answer appears to be yes. Groundbreaking work from labs like David Sinclair's at Harvard has shown that transient or temporary expression of some of these Yamanaka factors can reboot the epigenome of old cells, returning them to a more youthful state without erasing their identity. In a landmark study, they were able to restore vision in old mice with glaucoma by reprogramming their retinal ganglion cells. They essentially reset the epigenetic age of the cells, allowing them to regenerate their axons and function properly again. This was the first demonstration of reversing an aspect of aging by turning back the epigenetic clock. This field is in its absolute infancy and is still largely confined to animal models. The challenges to translating this to humans are immense, particularly concerning safety. The risk of inducing cancer by over-reprogramming is very real and must be meticulously managed. However, the potential is paradigm-shattering. Instead of targeting individual downstream hallmarks of aging, epigenetic reprogramming aims to reset the master program itself. It offers the theoretical possibility of not just slowing or stopping, but truly reversing the aging process at a fundamental level. This is the ultimate goal of longevity science. We've covered a tremendous amount of ground today, from foundational dietary strategies to the absolute cutting edge of gene therapy. The 10 approaches we've discussed represent the most promising avenues of research in the fight against age-related decline. While no single therapy is proven to dramatically extend human lifespan yet, these approaches are paving the way for healthier aging. They're moving us from a reactive model of treating individual diseases to a proactive model of targeting the underlying biological processes that cause them. It's a fundamental shift in how we approach health and medicine. It is absolutely crucial to reiterate that most of these interventions are still experimental. The landscape is filled with both incredible promise and significant hype. Therefore, I strongly encourage you to consult with a qualified healthcare professional before considering any of the interventions we've discussed, particularly the pharmacological ones. Self-experimentation can be dangerous, and what works in a mouse model does not always translate to humans. Your doctor can help you navigate this information and make decisions that are safe and appropriate for your individual health circumstances. The science of longevity is one of the fastest moving fields in all of biology. New discoveries are being made at an astonishing pace, and our understanding of the mechanisms of aging is deepening every single day. The journey to extend human health span is a marathon, not a sprint, but the progress is undeniable. Thank you for joining me on this deep dive into the future of aging. Stay tuned for more updates as research continues to evolve.